All right, the next speaker we have is Dr. Kathleen Houlahan, who is an assistant professor in the Department of Biochemistry and Biomedical Sciences at McMaster University, and serves as a principal investigator at the Center for Discovering Cancer Research, also at McMaster University in Hamilton, Ontario. Dr. Houlahan's work focuses on advancing our understanding of cancer biology, driving forward critical research that explores novel pathways and therapeutic targets in the fight against cancer. Dr. Houlahan was selected as the 2024 Canada Gairdner Early Career Investors by this year's Gairdner laureates, Drs. Balasubramanian, Klinnerman, and Dr. Mayer. Please join me in welcoming Dr. Kathleen Houlahan.
Thank you so much, and I just want to start by saying a big thank you to the organizers as well as the laureates that we're celebrating today for selecting me to give a talk today. So I'm really excited to tell you a story about how we're leveraging the technologies that we've heard a lot about to sequence DNA in order to uncover how our inherited genome may influence tumor evolution. So if we take a really big step back and ask what causes cancer, we know that cancer is caused by genomic and epigenomic aberrations, the causes of which can be broadly stratified into three categories: environmental, genetic, and stochastic, which here I'm referring to replication-associated mutations that just seemingly occur by chance. And these three factors come together to form tumors whose somatic mutational profiles actually vary quite drastically from tumor to tumor. But we know that we're all born with DNA that differs from individual to individual. And these somatic mutations occur in the context of that inherited DNA, which begs the question, does this inherited variation influence the somatic variability in tumors? And while this is unlikely to be completely deterministic, does this inherited variation actually start to bound the likelihood of specific stochastic alterations? And if so, how? One thing we know is that our germline genome can modulate immune response. So T-cell infiltration and cytotoxicity, as well as interferon signaling, is estimated to be about 15 to 20% heritable. And this is important in the context of somatic mutations because increasing lines of evidence suggest that avoidance of the adaptive immune system is a determinant in which mutations can persist in tumors. So these mutations in protein coding regions can generate novel peptides, about 9 amino acids in length, a subset of which can be presented on the tumor cell surface via MHC class I complexes. And these peptides and protein complexes, known as epitopes, a subset of these can be identified by cytotoxic T cells, which then eliminate the tumor cells. Now, these immunogenic epitopes are largely thought to be derived from abnormally expressed protein sequences or altered protein sequences. This is because our germline DNA should be considered self, and immune response to self should be dampened by tolerance mechanisms in order to avoid autoimmunity. But increasingly, we're learning that there are specific circumstances that facilitate escape from tolerance. So there's actually a similar number of peripheral cytotoxic T cells that can recognize foreign versus self antigens. They're just held in a suppressed state by peripheral tolerance mechanisms. And as I mentioned, specific circumstances can facilitate escape from this tolerance. And so this really ultimately led to the hypothesis that perhaps one mechanism by which our inherited DNA influences somatic mutations is by influencing immune responses. And ultimately, this led to the hypothesis that germline-derived epitopes in oncogenes may select against oncogene amplification. What do I mean by that? We're going to look at this toy example. Here we have individual A, who has a germline sequence of gene X. Gene X is set to represent a recurrently amplified oncogene in cancer. We're going to be talking about breast cancer, so let's imagine gene X is HER2, or ERBB2 that encodes HER2. Based off of this individual's germline sequence and their HLA class I alleles, let's pretend that there are two peptides that can be derived and presented from gene X. Now, if this individual develops a somatic amplification after unfortunately developing cancer, and we now have 3 copies of gene X, if each individual copy produced 2 epitopes and we now have 3 copies, 2 times 3 equals 6. We contrast this now to individual B, who has a different germline sequence of gene X and a different subset of HLA alleles. Let's pretend 3 epitopes can be derived from gene X in this individual. If this individual develops a somatic amplification of gene X, 3 epitopes per copy, 3 copies, 9 resulting epitopes. And you probably see where I'm going with this, but now we have individual C as contrast, which only has 1 epitope derived from gene X. After somatic amplification, 3 resulting epitopes. Now, if we take epitope availability as a surrogate for presentation likelihood and the likelihood that at least one of these might be immunogenic, we can see individuals A and B at an increased risk of undergoing immune-mediated cell death. And so in these individuals that start with a high epitope burden, somatic amplification comes at a fitness cost. And we can test this, leveraging the technologies we've heard a lot about this morning. We can look in retrospective patient cohorts and ask if there's a dearth of those individuals that start with a high epitope burden that go on to develop a somatic amplification. Okay, at this point you might be thinking, yes, but these are germline variants. How do we know that they're immunogenic? So as proof of concept, we first turned to the literature to identify non-somatically mutated peptides that were immunogenic. And we found 2, GP2 and E75. Both of these are derived from HER2. They are, again, non-somatically mutated, so essentially derived from the germline sequence, peptides that have been repeatedly shown to be immunogenic in various settings. And we hypothesized that the ability to present either of these peptides, and by that I mean have MHC class I complexes that can bind and present either of these peptides, would be negatively associated with developing HER2-positive breast cancer. This is because overexpressing HER2 and increasing the abundance of these proteins would trigger immune surveillance in the individuals that could present them. And so, like I said, we can test this. Retrospectively, in 630 primary breast tumors, we can look to see if having HLA alleles that can present either of these peptides is negatively associated with developing HER2-positive breast cancer. And that's exactly what we saw. So when we stratified patients based off of their HLA alleles and their ability to present either of these peptides, we saw a significant negative association with developing HER2-positive breast cancer. Okay, but these are just 2 peptides. So next we wanted to look systematically within ERBB2, which is the gene that encodes HER2. So we took the variant sequence of each individual, including all protein-coding germline variants, and predicted the epitopes that could be derived from the sequence based off of the individual's HLA alleles. And then we did the same thing with the human reference genome and essentially normalized the number of epitopes that could be derived from the variant sequence against the reference sequence, calculating what we're calling the germline epitope burden, or GEB for short, and just bidding patients based off if they had more, the same, or fewer epitopes derived from their variant sequence compared to the human reference genome. And then we can test if this germline epitope burden is associated with the likelihood of developing HER2-positive breast cancer. And here, to ensure that we're looking at a biological signal not confounded by differences in ancestry, we looked at individuals of European descent, but we're really excited to leverage the All of Us cohort and some of these other diverse cohorts to ensure that this generalizes. And we can do this systematically across multiple independent cohorts spanning all stages of breast cancer, but for the sake of today's talk, we're just going to be focusing on primary invasive breast cancer. And so when we look at this, we see a story very similar to the GP2 and E75 story, whereby individuals with a high germline epitope burden are significantly less likely to develop HER2-positive breast cancer. Now, HER2 is not the only focally amplified oncogene in breast cancer. There are actually 4 subgroups of ER-positive breast cancer that are defined and characterized by focal amplification of different regions of the genome, which is indicated here in this table. So we can essentially do something very similar to HER2, but this time calculating the germline epitope burden in the represented genes listed here, and ask if a high epitope burden is negatively associated with developing the subtype and then the somatic amplification that characterizes it. And so when we basically plot a bar plot very similar to the one on the left here, but take the odds ratio and now plot it along the x-axis, we can see that across these subtypes, a high germline epitope burden in each of the amplified genes is negatively associated with developing a somatic amplification in these regions. And I should note I showed 4 subtypes on the previous table, but 5 on the previous table and only 4 here, and that's just because one had too few missense mutations in the amplified genes to be sufficiently powered for this analysis. And this is showing an analysis in our discovery cohort, that's 630 primary breast tumors, but we see the same association and a replication of 3 individual cohorts, and this is showing the meta-analysis of those cohorts. And then finally, I just want to leave you, we can also look at sites of heterozygosity almost like an internal control. And so at these sites of heterozygosity, we might imagine one of 2 scenarios. Scenario 1, the reference allele produces an epitope with a higher binding affinity. If this is the case, we might imagine that the tumor would preferentially amplify the alternative allele in order to avoid immune detection. Conversely, we could imagine in scenario 2, the alternative allele produces an epitope with a higher binding affinity. In this case, the reference allele may be preferentially amplified. So in other words, we expect, if this hypothesis is true, to see an inverse relationship between the binding affinity of the allele and which one gets preferentially amplified. And when we look at a common variant within ERBB2, again, this is the gene that encodes HER2, that's exactly what we see. So when tumors preferentially amplify the alternative allele has weaker binding affinity. In other words, the allele with the weaker MHC class I binding affinity is preferentially amplified. And this is just one common variant, but we see the same thing in a second common variant in TUB-D1. And so really, what I hope you can take away from this today is this evidence suggests that the germline may be involved in immunoediting, and that a high burden of germline-derived epitopes in an oncogene is negatively associated with oncogene amplification. And in this way, we think germline-mediated immunoediting may serve as a novel mechanism by which our inherited DNA influences which somatic mutations a tumor develops. And so this was work largely done during my postdoc, so I have a lot of people to thank, but especially I want to thank my postdoctoral mentor, Dr. Christina Curtis, the whole Curtis lab, as well as our collaborators, Drs. Mike Angelo and Rob West, our funding sources, and I've just started my own group at McMaster University, so if this is of interest, please do reach out. Thank you so much.